Good day, everyone, and welcome to another screencast in Serology. I am your professor, Dr. Supache Bashid, and for today, we will be discussing serological and molecular detections of bacterial infection. Okay, so for this particular chapter, we will be discussing the host microbe relationship, the bacterial structure and virulence factors, immune defenses against bacteria and escape, laboratory detection of bacterial infection, and clinical manifestations and laboratory detection of selected bacterial infections such as the group A streptococci, Helicobacter pylori, Mycoplasma pneumoniae, and the febrile agglutination test, which includes the Widal test, the serologic test for Salmonella, and the Will-Felix test, the serological test for rickettsial. Okay, so let's review the host microbe relationship. So we all know for a fact that there are several kinds of relationships. So when we say symbiotic, the host and the microbes live together for a long period of time. So the most common example of symbiotic relationship happens between the human host and the indigenous microbiota. Now, in some cases, there are also relationships which can be categorized as commensalism or commensalistic, meaning to say there is no benefit or harm to either of to either organisms, whether we are pertaining to the host or to the microbiota. Now, when we say mutualistic relationship, both the host and the microbes would actually benefit. Okay? However, among the four types of relationship that we have enumerated here, the one that is the one that is considered to be as the worst relationship, or the one that is considered to be as significant when it comes to medical microbiology, that is what you call the parasitic relationship, because here the microbes can cause harm to the host. So that's the kind of relationship that we as human hosts would want to avoid, of course. So let's define first some of the terms that we are actually using in in order for us to further understand this lesson. So when you say infectivity, this is the ability of the microorganism to establish an infection. Okay, so whether some of them are overt pathogen or some of them could even be opportunistic pathogen, for as long as they are able to establish an infection. Okay, so that's what you call infectivity. Okay, so once they have established an infection, so perhaps one the second term that you'd be most interested with is pathogenicity, the ability of an organism to cause diseases or to cause disease for that matter. Now, why do you think they're able to cause disease? Because some of these microorganisms may have virulence factor within them. Okay? So virulence refers to the extent of pathology caused by an organism whenever it infects the human host. Okay? So that's what you call virulence. So there are several parts of the bacteria since we're talking about serologic detection of bacterial infection. So let us familiarize ourselves with some of the basic structure of the bacteria that may somehow contribute to its virulence factor. So one of them would be the endotoxin. So remember that not all bacteria are capable of producing endotoxin. So endotoxin is actually being synthesized in the cell wall of gram-negative bacteria. Now, chemically, endotoxin is made up of lipid A, so a portion of lipopolysaccharide in gram-negative cell wall. So why do you think it is considered to be a surveillance factor? Well, endotoxin is a potent antigen. In fact, in some immunology textbook that you might be actually reading, it is considered as the super antigen because it is a powerful stimulator of cytokine release. Okay? Now, on the external part of the bacterial cell, you will find villi. Okay? So in some texts, villi may be used synonymously with fimbriae, but actually villi and fimbriae are different. So pili are much longer, and physiologically they would do to see a different action with that of the fimbriae. So pili is actually responsible for the transfer transfer of DNA. Okay, from one bacterial cell to another, we call it conjugation. Now, a much shorter variety is called fimbriae, and that is the one that is important for adherence to host cells, which will make them resistant to phagocytosis. So, a good example of bacteria with fimbriae is Neisseria gonorrhoeae, the causative agent of gonorrhea. So, they have fimbriae, and this fimbriae will allow themselves to stick or adhere at the epithelial lining of the urethra. And because of the extent of that particular infection, so many of the segmenters, many of the neutrophils will try to go to the site of infection, and that will result into pus formation. This is the reason why a person with gonorrhea, particularly male patients, will experience urethral structure, hence the burning sensation whenever they urinate. Okay, so that is pili slash fimbriae. Then, of course, flagella. As we all know, flagella would be responsible for motility or locomotion of this bacterial cell. So they are also important for adherence to host cells. Okay, and then capsule, an organized polysaccharide, slimy polysaccharide layer found outside the cell, and of course, it allows the bacteria to evade phagocytosis. So it blocks phagocytosis, it blocks the attachment of antibody, and of course, it also blocks the the it also blocks the complement cascade. Okay, so which means that remember in the classical pathway of the complement, the C1 QRS complex is the one that binds at the cell wall of the microorganisms. So our complement pathway cascade, the switch of our pathway cascade will be much more difficult if the bacteria are actually encapsulated. And then exotoxin. Okay, so what's the difference between endotoxin and exotoxin? So by the way, we can find exotoxin in both gram-positive and gram-negative bacteria. So exotoxin is a potent toxic proteins being released from living bacteria. So that's one difference. So it's unlike endotoxin, it is embedded in gram-negative cell wall. Exotoxin is actually being released from living bacteria. Okay, so there are several variations of this exotoxin. It could be a neurotoxin. So it depends on what would be the target cell. So if the target cell is a derm cell, for example, so we call it neurotoxin, or we can call it cytotoxin, or enterotoxin if the target cell would be the intestine. Okay, so as of course we learned during the introductory part of immunology, we as humans are not really defenseless against this bacteria because we have both the innate defenses and adaptive defenses. So let's review. When we say innate defenses or natural defenses or non-specific immunity, so this may refer to the intact skin and mucosal surfaces because these epithelial barriers will actually bar microorganisms from entering our body. And then, of course, we also have antimicrobial defenses, peptides being secreted by our body, such as the lysozyme, defensins, and ribonucleases. And then, of course, the complement proteins, which will, once it is initiated, this may also contribute to cytolysis. Cytokines, although we do not want to have cytokine storm because this could also be harmful to our body. Then, of course, the production of acute phase reactants such as the C-reactive protein. And then, of course, in order to initiate phagocytosis, we will be producing toll-like receptors. Okay. And then, of course, aside from the innate defenses, we also have the adaptive defenses, otherwise known as the specific immunity. So these specific immunity could either be humoral response, wherein there is an antibody production by the plasma cell. So this will involve binding of complement, opsonization, neutralization of bacterial toxins, and the other one is of course the cell-mediated immunity. In cell-mediated immunity, lymphocytes such as the CD4 T helper cells would have the ability to produce cytokines, and that alone will be enough to induce inflammation. And as we all know, inflammation is very important because if there's inflammation, we prevent systemic infection. And then, of course, we also have the cytotoxic T lymphocytes. Okay, because of the perforin and granzyme. Perforin, of course, will cause hole in the target cells, and then granzymes will allow these particular target cells to be lysed. So again, when we say target cells, this may refer to virally infected cells or cancer cells. Okay, so this particular diagram here shows the ability of the neutrophil, which is the most when it comes to population, this is our most prominent leukocytes. Okay, so our neutrophil is capable of diapedesis. So diapedesis is a term that we're using wherein the neutrophil will squeeze themselves, okay, through the epithelial border of endothelial cells, the epithelium that lines the blood vessel. Okay, so in diagnosing bacterial infection, of course, the gold standard would be to isolate the bacteria by means of culture. However, not all cases that culture would be practical. So that's the reason why in the laboratory, there are several ways of diagnosing or detecting bacterial infection. So basic would be culture and microscopic examination. So when you say culture, we allow these organisms to grow in broth or solid media. Major means of diagnosis, but it may take time. In fact, for Mycobacterium tuberculosis, it will take about a month before you can have an isolated culture, okay, isolated colonies. Or in some cases, it may not be possible because some bacteria, the cultivation of some bacteria may be possible but dangerous. For example, rickettsia. Culture is dangerous. That's the reason why it's very rare that we are culturing rickettsia in artificial culture medium. It's not a routine procedure. And then Treponema pallidum, the positive agent of syphilis, it's very rare that we cultivate them in artificial culture. Microscopic examination will tell us a little story. It will tell us the morphology of the cell, or if we're using special stain, it will tell us whether the bacterial cell is present or not. However, it is not confirmatory, so it may help the clinician, but it is indeed not confirmatory. That is the reason why in some cases we are detecting bacterial antigen by rapid testing such as ELISA or fluorescent assay, labeled fluorescent assay. And then molecular detection of bacterial DNA or RNA can obtain results in few hours with, of course, polymerase chain reaction. So here we have now an example of bacteria that is detected using microscopic examination. So this is the gram stain. So you'll be able to see gram-positive cocci in cluster. And the illustration here is an example of an acid-fast bacilli. Okay, so stained with carbofuscin as the primary stain. Okay, so let's talk about serological tests. So in serological tests, we try to detect antibodies. So we try to determine whether the patient has antibodies to certain bacterial antigen. So it is used to detect and confirm infection for which other laboratory methods are not available. So it can also be used to diagnose infection for which clinical symptoms are non-specific. So current infection indicated by the presence of IgM and a high IgG titer. So current infection meaning meaning when you say current infection, the person is having an acute infection. So you'll have IgM, because IgM is the immunoglobulin for primary response, and then IgG is the immunoglobulin for secondary response. So a fourfold rise in antibody titer between the acute and convalescent sample is also considered to be as negative. Now, it may not be confirmatory, but having an antibody testing will also allow us to determine whether the patient has a past exposure to an organism. Let's say, for example, the IgM test is negative, but the IgG test is positive. So, this particular serological test will also allow us to assess reactivation or re-exposure to a particular bacterial antigen, especially if long-term immunity to a certain infection is not achieved. Okay, what are the disadvantages? Of course, the delay between the start of infection and production of antibodies. So there's this certain lag phase wherein there is no detectable antibodies, but it doesn't necessarily mean that that particular patient is already negative to a certain antibodies or low antibody production by immunosuppressed patient. So that is the reason why rapid antibody testing may not be feasible to all type of population. Okay, so let us discuss what are some of the common bacterial infection that we are using for for detection of uh, what are the most common serological tests that we are using for the detection of bacterial infection. That's what I meant. Okay, so we have here the group A streptococci. So gram-positive cocci arranged in pairs or chain, and this particular group A streptococci, such as streptoyogenes, is capable of person-to-person transmission. So, clinical manifestations of acute group A streptococcal infection will include pharyngitis or our strep throat, pyoderma or impetigo, scarlet fever. Do you remember this one? Strawberry tongue. Toxic shock syndrome. So it may also be caused aside from Staph aureus. According to Stevens, which is the reference of this book, group A streptococcus may also cause toxic shock syndrome, necrotizing fasciitis, and it is usually treated with antibiotics. So these are examples of streptococcal infections such as the strawberry tongue or scarlet fever. This one is, I think, is a pharyngitis, and then impetigo. Okay, so actually, group A streptococcus infection is really common. However, the one that we do not want to happen, especially if we leave it untreated, is the so-called post-streptococcal infection. This is the sequelae or the consequences of having group A streptococcal infection, such as the presence of acute rheumatic fever. So it develops 1 to 3 weeks after pharyngitis or tonsillitis in 2 to 3% of infected individuals. So acute rheumatic fever would have the following symptoms: fever, joint pain, inflammation of the heart. So most likely caused by immune responses to streptococcal antigens that cross-react with human heart tissue. So that's the reason why I never ignore a simple sore throat. Or if heart is not affected, so there's a chance that their kidneys may be. So this is what you call the post-streptococcal glomerulonephritis. It may follow strep infection of the skin or pharynx. So we do not want this to happen because in glomerulonephritis, there would be damages in glomeruli, and glomeruli would be important for filtration. And then eventually there would be hematuria, proteinuria, edema, hypertension, malaise, backache, abdominal discomfort, and impairment in renal function. So one of the reasons why this happens is because of the deposition of immune complex, okay, in the basement membrane of the kidney. So immune complex actually made up of antigen, antibody, and complement. So the antigen here is actually the streptococcal antigen. So this immune complex will be deposited in glomeruli attracting neutrophil and neutrophil, okay, will try to engulf this particular immune complex and damaging the glomeruli in the process. So that is the reason why this particular type of infection may be considered as the type 3 hypersensitivity reaction. So to diagnose group A streptococcal infection, we may culture the throat swab, for example, in sheep's blood agar, wherein there would be small translucent colonies, clear zone of beta hemolysis. Or in some cases, we can do the rapid assay to detect group A streptococcal antigens, such as the labeled fluorescent assay. Okay, so in some cases, we can perform the so-called laboratory rapid test, okay, for the laboratory diagnosis of group A streptococcal infection. So one of them is anti-streptolysin O, or the ASO. Okay. So before I discuss ASO, so let me point out to you that there are actually two types of streptolysin that is being produced whenever we are being infected with streptococcus. So we have the streptolysin O, and the other one is streptolysin S. So streptolysin O is oxygen's S. Let's talk about streptolysin S. So streptolysin S is oxygen stable, and it is responsible for the surface hemolysis, okay, on the surface of blood agar plate. So the problem with streptolysin S is that it is non-antigenic. Okay, so that's the reason why there's no such thing as AS or ASS. Okay, because it is non-antigenic. Unlike streptolysin O, although streptolysin O is oxygen labile, but this one is responsible for the subsurface hemolysis on blood agar plate. But relatively compared to streptolysin S, streptolysin O is oxygen is antigenic, meaning it is capable of eliciting antibody production. Therefore, whenever we get infected with streptococcus, group A streptococcus for this matter, okay, for that matter, there's a chance that our body would be producing anti-streptolysin O. Now, classically, anti-streptolysin O can be assay using the so-called neutralization test, wherein we're using the sheep's RBC as our indicator, and a positive result is no hemolysis. If you can remember that, the classical test for anti-streptolysin O, the reference range would be less than 165 TODT unit. Okay, so of course, it's a classical test and it's not being done anymore. The one that is actually more common in the hospital nowadays is the latex agglutination test, or ASO, wherein you can do the qualitative method and eventually the semi-quantitative method, wherein you will have serial dilution of the serum. And here in the semi-quantitative method, you'll be able to determine the titer of the antibody. So the reference range is less than 200 IU, which stands for International Unit per ml. Okay. However, the one that is being discussed in this slide is the nephelometric method. So this one is more sensitive as compared to the ASO latex. So the one that is shown here is an example of ASO latex. So the nephelometric methods currently used, okay, it measures the light scatter produced by immune complexes containing the streptolysin antigen. So the titer elevated in 85% of patients with acute rheumatic fever, and it does not increase in patients with skin infection. So it's more specific also. Because in ASO, the problem with ASO is that whatever form of your streptococcal infection, it will always yield high titer. So having a positive and high titer with ASO does not necessarily translate to acute rheumatic heart failure. Unlike if we're using the phelometric method, it is somehow more specific. But of course, clinicians would usually relate um, laboratory findings with that of the clinical presentation and history of the patients. Okay, so the anti-DNase B is produced by both rheumatic fever and impetigo patients. So it may not be really specific for rheumatic fever, and it is tested by enzyme immunoassay and nephelometric methods. So other streptozyme that may be useful aside from ASO is the anti-hyaluronidase. So hyaluronidase is an enzyme also known as the spreading factor. Okay, anti-streptokinase. So streptokinase is an enzyme that can usually lyse blood clot or coagulation. And this is an enzyme, an antibody against the coenzyme, the anti-nicotinamide adenine dinucleotide, and the anti-DNase A. So these are the different streptozyme tests that may use as or that may augment, okay, the ASO testing. Okay, next up we have the Helicobacter pylori. So it's another example of gram-negative microaerophilic spiral bacterium, and it is being transmitted by fecal oral route. So the major cause of gastric duodenal ulcer, the type B gastritis as what they call it. So as we all know, our gastric juice is highly corrosive. It's very acidic when it comes to pH. However, Helicobacter pylori can survive in acid environment of stomach because they are capable of producing urease, which will eventually neutralize the hydrochloric acid. So it provides buffering zone around the bacteria. So treatment with antibiotics and anti-ulcer medication would become the combination therapy for the treatment of this particular type of gastritis. If untreated, it can lead to gastric carcinoma or the mucosa associated lymphoid tumors. Okay, so that is the reason why it's very important that clinicians would be able to diagnose it right away. So it's quite difficult to culture Helicobacter pylori. Okay, so that is the reason why some tests would employ the detection of urease in stomach biopsy or the flow test. Okay, so here it detects the urease enzyme of Helicobacter pylori, which means that if you have a dark pink after the addition of the gastric biopsy materials, then it means that it is positive for the urease enzyme. So the other one is the urea breath test. In the urea breath test, you will literally breathe into this bug, okay, as part of the kit. Okay, so what does it contain? So the kit actually contains a C-labeled urea. So this particular urea will be if the person has Helicobacter, they will have urease. So urease will hydrolyze urea into ammonia and carbon dioxide. And since it is C-labeled, so the product would be also C-labeled CO2, and that is the one that will be measured in scintillation counter. Okay. And then, of course, detection of H. pylori antigen. So the one among these, of course, urea breath test is less invasive. So another one, we can also have the antibody detection. So in H. pylori antibodies, ELISA is the method of choice, wherein the presence of IgG in serum indicates an active infection. So eventually, your antibody titer will decrease after successful. Okay, so let's talk about Mycoplasma pneumoniae. Mycoplasma pneumoniae is an example of PPLO. So as we all know, PPLO are the pleuropneumonia-like organism. So this particular type of organism doesn't have any cell wall. So hence, if you will be looking at their morphology under the electron microscope, because they're so small, you would they are said to be there would be they're described to be as pleomorphic. Since, as we all know, cell wall contributes to the shape of the bacteria, and since these organisms do not have cell wall, they do not have peptidoglycan, so they won't have any definite shape. Hence the pleomorphic morphology. So, Mycoplasma pneumoniae is the leading cause of respiratory infection. So, this particular respiratory infection is characterized by fever, headache, malaise, and cough. Now, this one is also known as the walking pneumonia because in some cases, patients could be asymptomatic. So aside from this, patients could have the Raynaud's phenomenon. Usually, we can see Raynaud's phenomenon among patients with rheumatoid arthritis because of the constriction of small blood vessel. But because of the constriction of the small blood vessel, so the digits of the hands, okay, morphology would be greatly affected as shown here in the illustration. And in some cases, Mycoplasma pneumoniae may cause Stevens-Johnson syndrome in minority of cases. So how do we get Mycoplasma pneumoniae? Again, by means of respiratory droplets. So wearing of surgical mask is, of course, one way of preventing the infection. So how do we diagnose it? Culture. It's quite difficult to culture it. It's quite difficult to culture. There are commercially available selective medium for Mycoplasma pneumoniae infection or for any other species of Mycoplasma. So classically speaking, the culture medium for Mycoplasma pneumoniae was first developed in 1965. So it's kind of not true age, but it's kind of an old type of culture medium. It was developed by Neil Friis. So they actually call it Friis medium. Although there are now many commercially available culture medium for Mycoplasma pneumoniae. So it's still considered as a gold standard, but rarely performed in clinical laboratories because, again, it's quite difficult to grow. So that's the reason why many instead would opt to assay for the antibodies for Mycoplasma pneumoniae. So it's considered as the most useful diagnostic assay wherein IgM antibodies means recent infection, IgG antibodies means possible reinfection. Now, there is one test also for Mycoplasma pneumoniae. It's called the cold agglutinin test because apparently cold agglutinin is present among 15% of patients with Mycoplasma pneumoniae. It's not really popular because cold agglutinin is not really specific for Mycoplasma pneumoniae infection. So when you say cold agglutinins, RBC agglutinates optimally at 4 degrees centigrade, and agglutination disappears when you incubate the solution at 37 degrees Celsius. So the molecular method is the so-called film array respiratory panel. So it's actually a respiratory panel because it doesn't only detect Mycoplasma pneumoniae. In fact, it can detect up to 21 pathogens, including the coronavirus, although I'm not sure if it's the SARS-CoV-2, so perhaps they have already updated it, but when it was first developed, SARS-CoV-2 was not discovered yet, okay, or hasn't emerged yet for that matter. Okay, so apparently the film array respiratory panel has a very high sensitivity and specificity, more than 97%, and it doesn't only detect Mycoplasma pneumoniae, it may also detect for Bordetella, for example, respiratory syncytial virus, coronavirus, influenza virus. So it can detect up to 21 pathogens. So when they discovered it, SARS-CoV-2 hasn't emerged yet. So hopefully, they would already include film array respiratory panel. They would already include SARS-CoV-2 or the COVID-19, so that it's quite practical because you can easily rule out whether the upper respiratory tract signs and symptoms is indeed caused by COVID-19 or by any other pathogen. So that's the reason why I'm a fan of film array respiratory panel, but I'm not sure if this particular method is popular in the Philippines or available in the Philippines. Okay, so let's talk about one of the tests that is no longer useful. What are we talking about it? Okay, this is the febrile agglutination test. Okay, so the classical methods of febrile agglutination test, again, that's for the sake of undergraduate students, right? So let's define first what do we mean by febrile agglutination test. So it is caused by bacterial infection, and the reason why it's called febrile because whenever you're infected with this kind of pathogens, you are expected to have high fever. It's kind of vague because other pathogens will also cause you to have high fever. At any rate, this includes Vidal test for typhoid fever, the Will Felix test for typhus fever, the test for Brucella antigen, and the test for Francisella tularensis. For this particular lecture, we will just be focusing on Vidal test and Will Felix test. Okay, so Vidal test is of course the serological test for Salmonella. Okay, so Salmonella is the etiologic agent of typhoid fever. So we have four antigens of interest: O, H, and then we have the Salmonella paratype A and B. So O is a somatic antigen, H is a flagellar antigen, A and B are both somatic antigen of Salmonella paratypes. So actually, there are several ways of doing the without test. So of course, tube method is preferred over the slide method. Okay, and in the slide method, you can also do it in the without plate. Okay, so this particular test is an example of direct bacterial agglutination test. So in interpretation, if the significant titer is 1:80, which means that it has to be at least +2 to be considered as significant, right? So if you have more than 1:80 in O antigen, it indicates active recurrent infection. If you have more than 1:80 in H antigen, this indicates past infection, recent immunization, convalescent, or exposure. So a combination of A or B indicates paratyphoid fever. If O antigen is mixed with either A or B, this indicates mixed. Okay. However, nowadays, as what I've told you, without test actually lose popularity because of its very low specificity and sensitivity. So, majority of the hospital nowadays are using the typhoid test, consisting of a DOT ELISA kit that can detect either IgM and IgG antibodies against the outer membrane protein of Salmonella typhi. So the typhoid test becomes positive within 2 to 3 days of infection and separately identify IgM and IgG antibodies. Therefore, it will tell you whether you have an acute infection or you are on the road to recovery. So the test is specific. The test is based on the presence of specific IgM and IgG antibodies to a specific 50 kilodalton OMP antigen. So this particular OMP antigen is impregnated on nitrocellulose strips, and the nitrocellulose strips here serve as the solid phase. Okay, since the antigen is embedded on the nitrocellulose strip, so therefore the specimen is the patient serum, and the kit will detect patient antibodies such as IgM or IgG. So IgM shows recent infection, whereas IgG signifies remote or chronic infection. Okay, so it says here it is actually not time consuming. I forgot that. Okay, as compared to without test. So the most important limitation of this test is that it is not quantitative. So which means that unlike in Vidal, you cannot actually measure antibody titer. Okay? So since it is only qualitative, you either get a positive or negative test. So whereas in detailed without test, it can tell the titer. That's what I told you. However, typhi that with the lack sensitivity and specificity. So the without test is losing its value as it is labor intensive. So not all medtech should want to perform without test in the laboratory, especially nowadays. So if you will be comparing the sensitivity and specificity of typhi that and without test, so of course without test fail in comparison. So 86.2 sensitivity as compared to without 41.4 and 100% specificity as compared to without 91.4%. Okay, so when it comes to specificity, the best test among blood culture without typhi and diazo test, okay, when it comes to specificity, would be blood culture during the first week of illness, blood culture and typhi and test. But when it comes to sensitivity, the most sensitive would still be the typhi M test as compared to without blood culture or the diazo test. Okay, so let's talk about the Will-Felix test. The Will-Felix test utilizes Proteus antigen. Okay, so apparently, when Will and Felix discovered that person with rickettsia, okay, would actually excrete, okay, with they can actually detect Proteus antigen in the patient's urine of person with rickettsia. So that's the history of Will and Felix test. Can you imagine, no? So Will and Felix were able to relate the presence of Proteus antigen in the urine of a patient with rickettsia infection. So obviously, this one is not really specific for rickettsia. So nonetheless, when we say rickettsial infection, rickettsia is an obligate intracellular gram-negative bacteria. So an example would be the Rocky Mountain spotted fever, which belong to the so-called spotted fever group, and the other one is the typhus group, such as the epidemic typhus. So undergraduate students, typhus fever is different from typhoid fever. Okay, so the organisms are transmitted by arthropods, such as the ticks, mites, lice, or fleas, through biting after feeding on an infected animal. So these are the common rickettsial infection. However, let me point out to you that in this table, trench fever is not caused by rickettsia. The R there means Rocallimaya quintana, and Q fever is not caused by rickettsia. The C here represents Coxiella burnet. Okay, so these are the different organisms that may cause different rickettsial. Okay, so one of the most popular, of course, is the Rocky Mountain spotted fever caused by Rickettsia rickettsii. It is transmitted by 3 species of ticks, and symptoms include headache, nausea, vomiting, diarrhea, skin rash, and if left untreated, death. Okay, so diagnosis would be clinical presentation, of course, and indirect fluorescent assay. So it doesn't mention here Will-Felix test. So because Will-Felix test has poor sensitivity and specificity, that's why indirect immunofluorescent assay is considered to be as the gold standard. Okay, although it may be useful in investigating outbreaks or epidemics. So these are the reported results of the different diseases in Will-Felix test. So as you can see, a negative result doesn't actually tell you anything at all, because fever, trench fevers, rickettsial pox, and ehrlichiosis may yield negative results in Will-Felix test. So that's the reason why it doesn't really gain much popularity in the clinical laboratory. So indirect fluorescent assay is preferred, although this indirect immunofluorescent assay would be more expensive than the Will-Felix test, but it's more sensitive and more specific. That's the reason why it is considered to be as the gold standard. Not culture, because it's quite difficult or dangerous also to culture rickettsia. So Dr. Ricketts actually died from studying rickettsia. Okay, so that would be the end of our lesson for today. So again, this has been your professor, Dr. Subache Basit, saying, stay safe and God bless everyone.